Hello everyone, welcome to C Academy YouTube channel. My name is Veleleni Nkosi. In today's lesson we do the last part of meiosis. So we'll be looking at the importance of meiosis, abnormal of meiosis, and comparison of mitosis and meiosis. So this document that you see on your screen is an examination guideline from the Department of Basic Education. So since this document was written in 2021 and it's still active, so it's for life science, we will be doing meiosis. Meiosis falls under paper 2, which carries 21 marks. So 21 marks, it's about 7%. So 7% is a lot of marks. We have to make sure we get all these 7%. Without wasting more time, let's get to it. So the importance of meiosis. Meiosis, production of haploid gametes. So what do they mean by the production of haploid gametes? So during meiosis, haploid gametes are formed. These gametes have half the number of chromosomes from the parent cell. So if you can see this structure here, I have a parent cell and a parent cell which is the diploid number of chromosome, and then I have gametes, which is haploid number of chromosome. Gametes, when we talk about gametes, we talk about a sperm cell or egg cells. So this parent cell contains 4 chromosomes. So after the meiosis has taken place, we will end up with cells with only 2 chromosomes. This is what we call a haploid number of chromosome. Haploid number of chromosome is simply means the half the number of chromosome. So this one has 4 chromosomes, and then this cell contains 2 chromosomes. That is why they say meiosis produces haploid gametes. So haploid gametes just mean haploid cells. This will result in sperm cells if it's a male, and then if it's a female, they will end up with being egg cells. So this is the simple definition of this function. And then another function, meiosis. The importance of meiosis: the halving effect of meiosis overcome the doubling effect of fertilization, thus maintaining a constant chromosome number from one generation to the next generation. So here I will use this chart here to explain. As human, we have 46 chromosomes. Male have 46, and then female we have 46. If there were no meiosis taking place, so a male and a woman, when they conceive a kid, these kids will have 92 chromosomes. So that is mean if this generation have 46 chromosomes, the next generation will have 92 chromosomes, which is something that it's not supposed to happen. So meiosis makes sure that this does not happen. Meiosis reduce the number of chromosomes from 46 to 23 on paternal side. Again, will reduce the number of chromosomes from 46 to 23 from the maternal side. So sperm cell contains 23, and then egg cell also contains 23. When they fuse together during fertilization, 23 chromosome of sperm combined with 23 chromosome of the egg cell, we will end up with 46 chromosomes. So here is just the example I showed you, a pregnant woman, and then this side, the baby inside will have 46 chromosomes, which is the next generation from this parent. So it's also having 46 chromosomes. So this halving of meiosis make sure that each generation contain 46 chromosomes. Very important. This is another function of meiosis, or is the importance of meiosis. And again, something that meiosis do, the mechanism to introduce genetic variation through crossing over and the random arrangement of the chromosome at the equator. So genetic variation, it simply means we have different genes. We might be born from the same parent, but we have different genes. You remember we talked about crossing over, which takes place during the prophase 1 of meiosis 1. So this is the crossing over. The result of crossing over is when a homologous chromosome exchange section, this section of genes. Then each individual, this exchange is different. With someone, it might happen here and here. So it might happen also in the middle. It might happen like in the middle anywhere. So as long as if these are homologous chromosome, this exchange of gene, it might happen anywhere. So this is why most of the time we might be from same parent, but we look different, we do things different. This is the result of this crossing over. It doesn't have a formula of where it's happening. It just happens anywhere on the chromosome. And then another thing that increase this variation is the random arrangement. Random arrangement takes place during metaphase 1 and the metaphase 2. So in simple form, random arrangement is when these chromosomes are lining at the equator. Equator is this middle part of the sphere. So this chromosome, they might line anywhere. So there is no formula that tells if which chromosome should lie in which side. Now we go to the abnormal meiosis, non-disjunction and its consequences. So firstly, let me define non-disjunction. Non-disjunction is the failure of homologous chromosome or sister chromatids to separate properly during the cell division. So, and another thing, non-disjunction results in daughter cells with abnormal chromosome numbers. So I will show you a couple of examples here. This shows cell divisions. We have a normal cell division. So this cell has 4 chromosomes. But after meiosis has taken place, each cell here, the daughter cells, all of them, they have 2 chromosomes, which is the purpose of meiosis, is to have the number of chromosomes. So, but it might happen or an error might happen during the process. We end up with cells with more chromosome and some with less chromosome. So this process is the one that we call non-disjunction. We will end up with cells with a less number of chromosomes. So here the non-disjunction takes place during meiosis 1, and then here we have 3 chromosomes, while here we have 1. At the end we ended up with 1, but this side we ended up with 3. And then here non-disjunction takes place during meiosis 2, which is the anaphase 2. So meiosis 1 complete perfectly, but meiosis 2 did not do the good job. Here we have 2 chromosomes, here we have 2 chromosomes, which is perfect, but here we have 1 chromosome and then we have 3 chromosomes. I hope you can see this. This logo maybe is not disturbing you, but here there is 3 chromosomes, because here it's 1. This one from this side moves to this side. So this is the result of a non-disjunction. So this chromosome here, these chromatids here, they did not separate. They just went together to this cell and leaving this cell with only 1 chromosome. And then another important thing in biological term, when a cell contains 1 more chromosome than it's supposed to have, we call it a trisomy. It's a trisomy. And then when a cell has one less number of chromosome, one less, if it's supposed to be 2, you find that this cell has 1, and we call it monosomy. So these are very important. It's monosomy. So my handwriting is not the best one, but I hope you can see. This is trisomy. Yeah, this is this is I, here it's I. And then monosomy, it's when a cell has one less chromosome than it's supposed to have. Here it has one more chromosome than it's supposed to have. So this process here, we call it non-disjunction. The result of non-disjunction, we end up with a cell with abnormal number of chromosome. So this is the consequences of a non-disjunction. So another thing, non-disjunction of chromosome at position 21 during anaphase in humans to form abnormal gametes with an extra copy of chromosome at 21. So here we need to know when this non-disjunction takes place. So it happens in anaphase 1, where there may be non-disjunction of homologous chromosome into separate chromosome. So during the anaphase 1, homologous chromosome must separate. So one must go to the other side and one must go to the other side. Then it happened that they do not separate. So they are homologous chromosome and they are not supposed to be in the same cell. But it happened that they do not separate and they goes to the same side. So this is what we call non-disjunction happening at anaphase 1. So which is meiosis 1. And then it can also happen in anaphase 2 when there may be non-disjunction of sisters chromatids. So meiosis 2 takes place in two daughter cells. So here I have daughter 1. This non-disjunction happens in this cell. Instead of the sister chromatids separate, they do not separate, they went to the same side. So if you can see here, the chromatids separate perfectly, but here they do not separate. So here is when the non-disjunction takes place in anaphase 2. So you must be able to differentiate them because they will show you diagrams. You must understand these diagrams. So here it happened in anaphase 1, meiosis 1. This one, it happens in anaphase 2. So this is very important. And when these things happen, they will force a haploid karyotype. This is a haploid karyotype. If you can see that the chromosomes are not in pairs, so we call it haploid karyotype. But if you can see, this 21 chromosome contains 2 chromosomes. It's the result of non-disjunctions. So you must be able to read this. As human, we have 23 chromosomes, and the 23rd chromosome are the sex chromosome. So X is for female, while Y is for male. So that's why I put OR here. These are the sex chromosome. X is for male, X is for female, and then Y is for male. This is the result of non-disjunction chromosome. So we have extra chromosome which is 24 chromosomes. And then another thing, you must know the fusion between an abnormal gamete, which is 24 chromosome, and a normal gamete, which is 23 chromosome, may lead to Down syndrome. Here I have this gamete. This is a sperm cell. This sperm cell have 24 chromosomes. So disjunction, this process, this era of disjunction, non-disjunctions, it takes place in a male. So we ended up with 2 chromosomes on the chromosome number 21. We ended up with extra chromosomes. So we have 24 chromosomes. And then in female, on the X cell, everything is perfect. And then when they fuse a sperm cell and then an egg cell, we end up with a cell with 47 chromosomes. So if you can see, we have they are in pairs now from maternal and then paternal. But if you see this 21, we have 3 chromosomes. This is the result of this chromosome, this 21 cells, 21 chromosome. We ended up with 3 chromosomes here. So this we call it trisomy 21. That is mean there is an extra chromosome on chromosome 21. We call it trisomy 21. And then the result of this trisomy 21, it's a Down syndrome. So Down syndrome is a condition of people who like here I show you this doll. People with Down syndrome, they have a flat nose bridge. So this bridge here between their eyes, it's almost flat, some it's even flat, and then they have small eyes and then small ears. Even their nose, sometimes you find it's small, and then they have very short necks. So these are the people with Down syndrome or trisomy 21. You might call it Down syndrome or trisomy 21 when this human karyotype here have 3 chromosomes. So this is the result of one with 24 chromosome and then one with 23 chromosome. We'll end up with 47 chromosome, and then this extra chromosome is on number chromosome 21. This is, you must know the trisomy 21 or the Down syndrome. What causes the Down syndrome? We don't have to know this flat nose or small eyes or whatever. You just have to know that if there is an extra chromosome on number 21, the result of it is trisomy 21 or Down syndrome. So, and then going forward, now we will compare the mitosis and meiosis. First, let's start with the similarities of mitosis and meiosis. The similarities of mitosis and meiosis, both are form of cell division. Yeah, both are form of cell divisions. Both occur within the nucleus of the cell. They take place in the nucleus of the cell. They are through the same step known as prophase, metaphase, anaphase, and a telophase. So they go through these steps. Both of them, they go through these steps. It's just that meiosis goes these steps twice, while mitosis, it only takes place only once. So another thing, now we have to explain the difference between meiosis and the mitosis. So mitosis takes place within somatic cell. Somatic cell is a body cells, and then meiosis takes place within sex cell. So this is the difference. This one takes place to somatic cell. This one takes place to sex cell. And then no pairing of homologous chromosome. In mitosis, there is no pairing of homologous chromosome. And then in meiosis, pairing of homologous chromosome in prophase 1. So in prophase 1, there is pairing of chromosome, of homologous chromosome during meiosis. And then 2 daughter cells are produced. In mitosis, we ended up with 2 daughter cells. In meiosis, we ended up with 4 daughter cells. And then complete after 1 division. Complete after 2 divisions. Meiosis complete after 2 divisions, while mitosis only complete after 1 division. No exchange of DNA between the chromosomes. No crossing over in mitosis, and then in meiosis there is at least 1 crossing over between the homologous chromosome. As we talked about, the crossing over in meiosis, which takes place during the prophase 1, and then the number of chromosome per nucleus remain the same after division. Yes, in mitosis, the number of chromosomes remains the same. And then meiotic products contains haploid number of chromosome in contrast to the diploid number of chromosome in the mother cell. We know that the function of meiosis is to halve the number of chromosome from the parent cell, and then so it produces haploid gametes from the diploid cells. This is the function of this meiosis. So in mitosis, the number will remain the same. In mitosis, the number will be halved. In human, we have 46 chromosome. We ended up with 23 chromosome. But mitosis, 46, we still got 46 after the cycle has completed. So this is all for today. If you have watched it to this far, thank you very much. Please consider subscribing so that our channel can grow, so we can do a lot of research and give you good content. So if you are studying, I say good luck with your studies. Thank you very much.